For people off-site, we've only got 2 people on-site, so finally off-site attendees outnumber on-site. So if you guys want to just start yelling invectives from off-site, or whatever you choose to do, I'm sure you can kind of take over. Okay, so let's see. So the point of this week was to finally kind of hinge from, we previously did a lot of, generalized linear models, generalized linear mixed models to get going in TMB in the first 2 weeks, and then we did, time series models to get used to sort of correlation structures, and then, obviously did 1D spatial models last week, which is sort of an artifactually simple case for most purposes. So today's finally when, this week is finally when we get into 2D spatial models, and it's structured where today we'll do 2D models on an equally spaced grid, and then on Thursday, I'll be out of town, but Jay will be presenting a lecture on using the SPDE method, or kind of unequally spaced locations in 2-dimensional space. So that SPDE method, as you'll see, is what's incorporated, what was developed by the developers of INLA back in 2011, and so there's been this sort of growth industry of trying to do, spatial temporal models, kind of following this development that INLA pushed. And so by the end of the week, ideally, people will start to see how they can start doing kind of INLA-style models in TMB. But before we do that, I want to kind of build up to it from the tools that we already have in hand, and so build up specifically from these sort of first-order autoregressive models that we've been doing in time or 1D in space. Maybe we can do some brainstorming for a second about why we would care about 2D spatial models. Set some problems that this would open up.
Well, space isn't one-dimensional.
Yeah, that's good. I mean, yeah, in like. Sure. Oh, we share my screen. Yeah, of course. So I started recording. I always forget to do one or the other. Thanks for reminding me. Yeah, so there's a lot of things that are not. I mean, like the world operates kind of in space and time, but it operates. You can think of it as kind of being infinite dimensional or really high dimensional, and we are used to projecting things from high dimensions to low dimensions. And so there are cases where 2D space can be projected into 1 dimension, and there are cases where it can't. So that, obviously this opens up a set of spatial models where you can't project something like a coastline down to 1D. But what specific types of ecological or health questions can we address with kind of 2D spatial models?
Feel like this answer, like anything, yeah.
I mean, that's definitely my thing. Yeah, I mean, once I started doing spatial temporal models, I started seeing spatial models everywhere. But I would like to push us to actually come up with some concrete examples that we can talk about stuff during the course of class. So it's like Grant, you've been doing stuff with spatial overlap, right? And of course, you could project that down to 1D. You could say, like people do that. People have been doing this for a long time, where they say, a species has a certain distribution. They project it onto temperature, and then they look at sort of the overlap in that one dimension, in that projected space. How would that compare with doing it in 2-dimensional, like latitude, longitude coordinates? Do you have any kind of hypotheses coming in, how those approaches are different? I don't really know either. Yeah, I mean, you're probably losing some information when you project from 2 dimensions down to 1. But what the impact of that is, I have no idea, really. I doubt anybody does.
I feel like there's also more logical ways that you can talk about things that are related in 2D space, because then you have both, you have sort of directionality, and you can say things about whatever the spread of fish,
totally,
tends to be within this radius or something like that. Whereas with temperature, it's like, what does that mean when you find relationships or ranges?
Totally, yeah. Like in fish, we often have these sort of, like due to geology, like the continental shelf, as I understand it, like continents are made out of a different like geological material that floats higher on the mantle. And so like continental shelves are the same material, and so they are kind of close to the same surface, and then they drop off rapidly under kind of geological changes. And the kind of, so like when, those sort of depth changes cause things to be correlated really strongly across a given depth band and decorrelate really quickly as you go into deeper water. And so you end up with these sort of complicated correlations where, it might be really strongly correlated one way and weakly correlated another. It's easier to talk about that in 2D, I think. But like in public health, I imagine there's all sorts of like interesting, like income is probably correlated in complicated ways in 2D space, like along river. I don't know how you guys do this.
Yeah, there's all sorts of like covariates that are inherently spatial, like air temperature or
Totally
income or really anything.
Rainfall.
Yeah, but like a lot of those, like I think as we've talked about in our previous classes, our models have the impact of covariates that are included via fixed effects, and so we're talking about the kind of residual processes that we're not otherwise modeling. And presumably those residuals also have kind of complicated spatial patterns that are easier to pick up in 2D than projecting it into one dimension.
But ideally, couldn't you reduce 2D spatial models to 1D if you had perfect information on what drives variation in the population?
Yeah, maybe. Yeah, exactly. They presumably there's some problems where if you had perfect information, you could project it to a lower dimensional space. And and we're doing that all the time too. I mean, like in reality, like maybe there's like 3 or 4 dimensions. Like in water, it's easy to imagine there's a vertical distribution, but in public health, it could be the 3rd dimension could be age measured continuously, or income, or other kind of cohort or other continuously valued effects. And we could end up wanting to have a higher than 2D smoother, and we're always projecting that down to like, well, maybe 2D or on a kind of a small set of other covariates. That we're getting into kind of complicated stuff here, but I guess my claim is that this stuff that when I'm thinking about these, I always kind of try to keep in my mind that other people besides me actually do experiments, and so like the history of mixed effects models, as we've talked about, comes from kind of at least one branch that comes from Ronald Fisher, and doing these like randomization designs where there's a component of variation that he could not control. He had an agricultural plot, and you'd have a treatment and a control next to each other, but some of the variation in this experiment was random due to uncontrolled differences in soil quality. And so you could have an experimental model that has a experimental design where you've got some residual spatial variation that's worth modeling, and if you model that, you get better efficiency for a fixed set of fixed sample size. And then there's the observational analysis, like we're already talking about. One bandwagon that I think unites ecologists and public health people right now is sort of this emphasis on species distribution models. So can anybody define a species distribution model? Or
I think it's just any sort of statistical model that tries to evaluate how some, indicator changes across 2-dimensional space.
Yeah. Has anybody followed sort of like the literature about Maxent? Or
Isn't it just like a funky logistic model?
Yeah, but so like specifically there's sort of this, like in the mid-2000s, Jane Hewitt and others started doing these sort of models that were just fitted to presence-only data, quote unquote, so like instead of a logistic regression where you have presences and absences, she and others saw that if you just have records of presences, you could fit this model using maximum entropy, which is kind of like maximum likelihood. And so presence only models might make sense in some public health contexts if you just have people self-reporting, Zika, but you don't actually know who sampled themselves for Zika and doesn't have Zika because they don't know to report it. So like a lot of emerging public health issues, I think, end up being a presence-only model until people do kind of data recovery to start figuring out where the absences are. Anyway, so this sort of literature grew up about SDMs, this kind of modern literature about SDMs. And people have kind of progressively worked out that, a presence-only model like Maxent is really the limit of like a logistic regression under certain assumptions. And so these days, I just kind of come along and call species distribution models any model that projects, distribution, so presence, absence, in two dimensions, kind of like your definition. Some people want to define a species distribution model as presence absence, and a species density model as, modeling numbers, and then some people model biomass, and so forth. Anyway, I think a lot of people, from public health and for fisheries, were sort of interested in this Grinnellian niche. So anybody want to summarize sort of like Grinnellian versus Eltonian niche? Maybe this isn't a term that you guys use. My understanding is that Grinnell was an ecologist who sort of claimed that distribution was driven by sort of habitat, whereas Elton, Charles Elton was interested in phasian ecology and kind of imagined that distribution was controlled by kind of trophic, predator-prey competition, kind of biological interactions. And so people sort of refer to the Grinnellian niche as sort of the niche of sort of measured habitat variables and how they affect distribution, whereas an Eltonian niche would be some, way of modeling distribution based on the overlap of predators and prey and competition and explicit species interactions.
Why do they have to be exclusive?
Yeah, and then there's a bunch of tree papers where they're like, well, it could be both, and what if you model, like meta-community models, source sink dynamics, species movement and dispersal. But I think people kind of like having these terms as sort of cartoons of sort of the simple cases of the extremes. And I kind of like using it because a lot of our models that we're going to be looking at are very simple in the sense that they have a covariates, they have measured covariates like rainfall or income that, in the model are assumed to affect local prevalence or incidence of the disease. And then you've got a spatial residual term that's kind of naively interpreted as sort of, residuals of the prevalence of that disease. But, my feeling about the GBD is that, GBD is that, it doesn't explicitly model dispersal. It doesn't model if you have influenza, how far do people move in a week where they're contagious, or, if it's HIV, they don't model airline pilots spreading it between airline nodes. There's not a lot of explicit, or interactions between diseases with the same vector. So I think GBD is still in the kind of Grinnellian niche world, and a lot of our regression models are also still kind of essentially this kind of extreme of kind of a simple regression that's interpreted as a Grinnellian niche anyway.
With these climate envelope models, is that just pretty much you fit a model to some area and then project it into some other area?
I'm actually not an expert on how people use these terms. That's sort of how I use it, a climate envelope model being a distribution model that people use to project often into new climate scenarios.
I'm thinking of who's that guy from Canada?
Well, there's William Chang,
yeah,
Malin Pinsky both do these sort of, totally. And William's stuff with William Chung, William Chung is now, like leading the fisheries subgroup at the International Panel on Climate Change. You know, so he has a lot of influence in how we're thinking about climate impacts, you know, based on his work and his role. You know, and his stuff with Daniel Pauly, I think would have these very simple sort of like assumptions that like increased temperature decreases growth, and it affects some assumed logistic local growth rate, and then they just like assume that work explains all biology and project it. And you can probably tell from my tone that I'm sort of skeptical about how well that works. But
Yeah, and they call it a climate envelope model.
yeah, I think they do. So I mean, I think a climate envelope model is sort of like one of these species distribution models that, you know, typically has a fairly small set of kind of climate variables, you know, and assumes that there's no residual spatiotemporal process except for the kind of model regression that they're doing. The Williams model is a little bit even more confusing, I think, because he has like sort of non-linear biological effects, but it's not clear what he's fitting them to. So it's not even a regression exactly. It's sort of like a hard to, you know, interpret non-linear thing. Yeah, I don't know. I wish I knew more about how the GPD is working these days. Like, if you can you help talk about an example for a second about how?
Most of it's not geospatial.
Yeah.
Like, well, the GPD like explicitly isn't geospatial. Everything's on like the country level, so
Yeah.
it's very like discrete, and like the distance is measured in like a hierarchical like region, super region sort of like relationship. But the geospatial team, you have a geospatial team, so you could probably talk more about.
But in the non-geospatial world, it's sort of like there's like a regression relationship that they establish between income and prevalence of malaria.
Sure.
Yeah. And then they assume that they project off that relationship to predict into kind of areas without data or something. Is that?
Yep.
A cartoon?
Yeah.
Or a cartoon. Okay. Yeah. And the geospatial people are doing something else.
Yeah, well, we do like explicit, you know, 2D modeling, but I don't know if it would be called species distribution modeling the way you describe it, because we always have like a population, so there is sort of like a denominator of, you know, how many diseases there could be.
Totally.
Whereas it sounds like with these models, with like Maxent, it doesn't sound like there's any sort of like upper limit on the number of species that can be found within a certain area.
Yeah, so people do it differently. Yeah, like a lot of times people have like a Poisson, like a log-linked Poisson regression for the density, you know, the number of individuals in a species or the number of species. But you can, of course, kind of expand that to do like a binomial or something where you pre-specify the denominator of that binomial distribution. So I don't know. I mean, I don't mean to belabor the point. I'm just trying to get people thinking about sort of different classes of models and how they describe it. Here. And then, let's see. So, you know, I got interested in this. You know, so like I think last week at some point I talked about sort of like getting into Gaussian processes from this sort of like semi-parametric modeling where I'd want to have a functional relationship but model sort of patterns that are not explained by that functional relationship. And you know, my friend Oli Shelton, who works at the Northwest Center, you know, recognized that, you know, as a lot of people have, that a Gaussian process you can also do in 2 dimensions, and suddenly you get sort of, you know, residuals around the effect of covariance in a spatial model. So that's sort of the way we're talking about geospatial regression. So in this paper, which I love because Blake Feist, the co-author, among other benefits, he made these beautiful graphics. So we fit a spatial model to fish density from the survey one of the surveys that I think we've looked at. The one that we looked at canary rockfish. And in this case, we looked at dark blotched rockfish. And the way we did it before was we would spatially stratify the model, so maybe kind of like the way that the non-geospatial GPD works. You know, we stratify it based on depth. And so there's sort of shallow, mid, and deep waters. And this sort of spatially stratified model is what we use to expand up our, you know, calculate total abundance across the coast for our fish every 2 years when we do a cycle, kind of like the cycle we were talking about, the And so if we did this, using our old software that Eric Ward and I did, we found that this dark blue was lowest density, offshore and inshore had kind of slightly higher density in this light blue, looking at the scale, but the highest density was this sort of beige in the middle, where the location of sampling are marked with these circles or crosses. But if we instead went to this spatial model where we have a kind of quadratic effect of depth and then spatial residuals around that parametric relationship, we were able to pick out a depth band that had extremely high predicted density, so as high as like from 3 to 70 or something, shown in red. But it captured the kind of general low density offshore and onshore, inshore. And basically by doing this, it partitioned more of the residual, it modeled more of the variation in the data, within this kind of within stratum variation that it was modeling. And so the residual error term was lower in the spatial model than the spatially stratified model. And because the residual variation was lower, it meant that the predictive variation to new locations was lower. And so when we sum across space, we end up with a lower predictive variance for the sum. And so we'd end up with a lower standard error, a lower uncertainty when predicting total abundance in this model than this one. So that was sort of the hook that got people on the West Coast, I think, interested in these spatial models for our assessments. Okay, so as I said, I think we eventually want to get to this sort of unequal placement of locations that we're modeling, and that's sort of what inline kind of got people excited about doing, I think. But we'll start out with equally spaced locations. As a kind of side note, there's a whole other kind of subset of spatial models called aerial models. So these are all kind of like point process models where you say that you're modeling a location in space, well, a set of locations in space. Kind of a different approach that people developed independently, and it has some kind of deep connections, is to model areas, like countries, presumably, like in your geospatial stuff, if you're doing aerial models where you model country residual, errors at the country level, or are you modeling fine scale within countries? Or?
We do within countries, and so anything that we get that's like an aerial unit, we end up resampling the points, so it's all sort of comparable.
Yeah, okay, so that's good. What's that?
There were teams that were looking at like U.S. counties and
Totally.
modeling the spatial correlation between counties.
Yeah, okay. Yeah, I mean, so there is sort of this long tradition of kind of doing some analyses as aerial models where you say that like this county is adjacent to 3 counties next to it, and this one's adjacent to 5 counties next to it, and you have the partial correlation is restricted to counties adjacent to you.
Just a big old car model.
A big car model. Yeah, whereas point process models used to be kind of follow a different path. And then eventually people realized that, you could approximate point process models using a car model, and you can approximate aerial models using sort of a SAR model. And so in the end, there's not that much distinction between the two of them, except that there's sort of this tradition of different vocabulary that I want you to be aware of, but we're just going to think about point process models in the remainder of the class, really. Okay, so for an equally spaced grid, one example that, I mean, again, like if you guys had good public health data sets that were public, I would love to bring them in more. So feel free to email me, and I'll try to integrate them. But for now, this is a public data set that I scraped off the ICES website, which ICES is sort of the International Council for the Exploration of Seas, is a over 100-year-long organization that coordinates data collection for the Northern Europe for ocean stuff. So in this case, there's a bunch of different samples that have occurred, and if I define a sampling area, a domain of the sampling protocol based on the location of samples, I could get a bunch of black points here, and here's a red grid, where the red points are points that are far enough away from the samples that we have that I assume they're not within the sampling domain. And this is a grid that then I project using UTM back to latitude longitudes. That's why it doesn't look square anymore.
Never mind.
So this grid I did in Universal Transverse Marcator, which is supposed to be kind of like a distance preserving metric. And so obviously as you go north away from the equator on the globe, different longitudes are closer together. One longitude is a smaller distance than one longitude difference at the equator. So that's why I projected it into UTM, make a grid in UTM, and then project it back to lat long, just to kind of preserve distances a little bit better. Do people want to talk about projections more? I also don't really talk about projections that much in the class.
Yeah. I feel like projections are such a big thing in like most, especially like fisheries, like most people completely wipe over them.
Yeah.
I guess like moving between like WGS 84 and like UTM, you're only gonna be off like by like a few meters or something.
Yeah.
But
it depends on how big of an area you're using. And
yeah.
That kind of thing. Yeah.
That was what I was gonna ask, especially with an area that's like, I think, is it UTM that has like the 18 different zones?
Yeah, or I think it's 36, but whatever.
Okay. Yeah, if you're doing like a spatial analysis where you're extending them, into multiple UTM zones, what like projection would you use so you're still maintaining like?
Yeah. Honestly, for all my projection stuff, on the one hand, I don't talk about it that much because it's sort of stuff that used to come up in, like, Miles Logsdon would teach it in his, like, spatial information systems class. And, I think that there's sort of like a separate kind of literature of GIS stuff that ARC does that doesn't have that much stats and doesn't have that much theory, and so I'm just not that interested in it. Like, for my optics, it's sort of bookkeeping stuff, but I probably should have a little more of it in the class. And yeah, UTM definitely, divides the globe into 36 zones, and you have to keep track of whether it's northern hemisphere or southern hemisphere. And, falls over, the errors accumulate really quickly if you're projecting from multiple zones, like you're saying. And so maybe this is already kind of hitting the limit of what you probably want to do in terms of areas for UTM. And for most fisheries analyses, for surveys, we never really get a bigger area than this. So, my claim is that UTM is sufficient for a lot of our surveys that we do. For fishery data, like across, for pelagic tunas, we get the whole Pacific Ocean, and UTM falls over for that all the time. And so they end up doing a lot more detailed projections. There's a package called RGDAL that has like CRS, and I'm trying to think what that means.
Coordinate reference system.
Coordinate reference system. Yeah. So, if you can use software to use CRS projections where you have to provide like an alphanumeric code. And I don't really know that. Again, or the other people who use it in fisheries a lot are people who work near the poles, because UTM just works like shit near the poles. So like New Zealand uses CRS routinely, whereas our North American people, I don't think, can get into this as much. And then of course, if you're doing actually like a whole globe, there's, inlet has a bunch of features. You can define a car model, so like an aerial model of adjacency. You can define that actually on a spherical coordinate system. So inlet has a bunch of capabilities for that. And so in theory, we could do all the same stuff. We could do stuff directly on a globe if you actually wanted to model, like a GBD, if you wanted to model a globe where, you want correlations to actually wrap around the Earth in a bunch of different ways. I don't know if that's something that really comes up.
Well, this is super interesting because I was a GIS undergrad, and we do like Africa models, and I have not once thought about like the coordinate reference system we're using. So
yeah.
I feel like that would be a pretty big deal. So I'm gonna go back and try to figure out exactly what we're doing. I think we're just doing lat long, which seems really wrong.
Yeah.
Well, yeah, right. And again, So there's a paper by Daniel Simpson, who's Simpson's one of the people associated with Inla these days. And Daniel Simpson has a paper that titled something like "Why We Shouldn't Care About Correlation Functions" or something. So I think like GIS people care about projections a lot, and then geostatisticians care a lot about like, oh, you're using a spherical or exponential or Matern correlation function, and they spend a lot of time on both those questions, and they're kind of similar. I think as an applied ecologist and statistician, I'm so used to having messy data sets where I've got all sorts of biases in my analysis because my model is mildly misspecified in a zillion different ways. And so I kind of, am usually happy to just show that my results are similar using, a couple different correlation functions and a couple different, projections. And then I just assume the bias resulting from that is of similar, smaller magnitude than other biases. So. And I think that's why a lot of applied people don't stress about it as much as sort of the classic GIS and classic geostat crowds did. But it's, I'm sure it's worth every community having people that are chasing down those questions of like, is it sensitive to projection? Yeah.
Worth knowing, worth at least trying.
Yeah.
What was the name of that paper? It's just called "Why We Shouldn't Care About Correlation Functions."
Yeah, it's pretty close to that title. Yeah, and it's Daniel Simpson. And it's from like 2013 or something, somewhere in there. Yeah. So okay, so how do we actually want to do this on a 2D grid? Well, the code I'm going to show, again, has 4 different ways of doing it. But before we get there, I kind of want at least one of them to be sort of somewhat intuitive. And so we'll start out with some kind of notational stuff. So if we've got epsilon for row I and column j, then we can pull out a row of that, epsilon I being a row vector, and because it's a vector, I'm going to use bold, lowercase, non-italic. And then finally, if I want to refer to the matrix, I'll just call it E, so using vector matrix notation, use an uppercase, bold, non-italic. So E has 9 elements, a 3x3 matrix. Epsilon I is a vector of length 3. And going back a second, if we are doing this, we did this in time series case, and we did it in this 1D spatial case. We can define, according to the axiom of conditional probability that we defined sort of the first day of class, the claim is that we can decompose the joint probability of epsilon of just a row of this, or a 1D spatial process or a temporal process. We can decompose that joint probability into a recursive process, or a series of conditional probability statements. So we can describe the process both of these different ways. And this one I call kind of sweeping upstream to downstream often. It has this sort of recursive definition of conditional probabilities. And the joint probability, we can kind of work out by hand, looks like this, where, if they're 2 apart, they have a correlation of rho squared. And so we saw before that that's equivalent to defining exponential correlation function, in the unequally spaced case. And where this is that this epsilon squared is the same as this one. This is the marginal variance of the process, and it has this 1 minus rho squared in the denominator because it's this solution to a convergent power series as long as rho is between -1 and 1. So that's sort of recap of what we've talked about through week 5. And the final recap is that we also know that the inverse covariance matrix, or the precision matrix, is sparse, where it has a partial correlation matrix that indicates that everything is 0 except for the adjacent locations. So this is sort of like a class. This is sort of analogous to the CAR, the conditionally autoregressive specification of what was originally developed for aerial models, but extends to point process models. Okay, so the goal is to kind of get people thinking about how you would extend that upstream to downstream conditional specification for a 2D process. So if you were gonna, again, and this goes down to the kind of projecting 2D onto 1D, or how would you do that? Like, get any ideas about how you would define the joint probability of all 9 of these points, given the kind of tools at our disposal?
Well, you can. I mean, that previous car model matrix should be able to handle it. You just make it multidimensional.
Yeah, so one way to start doing it is to think about it as a car model. So like this point definitely has a partial correlation on this one, and this partial correlation should be 0 of this point to this point. Similarly, it should be 0 for these. Like the car model, like the partial dependence, I have trouble thinking about because I always get confused. Like, is the partial correlation 0 for this one, or is it non-zero? And so that's why I don't usually derive it that way when I'm thinking about it myself. If John were here, I'm sure he'd have some. I'm sure he's seen this before, so he would have some interesting thing to say about apply that idea to it. But any other ways that, like, using the kind of vocabulary we've used that people can think of?
Could you also think of it as like an additive linear process, I guess, or like I'm just thinking like break it up.
Totally, right? So you could think of it as, you can always think of any model, like there's like a kind of culture of like referring to nested versus cross random effects. And so you could think of this as like the marginal effect of rows. You could have a random effect that's just a 3-length random effect. Then you could have a 3-length random effect for this columns, and then their interaction, which would be called like I don't know if it actually would be called nested or cross. I always find that terminology confusing, which is why I don't use it. But you can define an interaction. But that one's also kind of hard because I don't know how then you just have to figure out what the correlation is for the interaction. I guess the claim, for me, I forget 2 years ago when I taught this what form I wanted to take for work through on the board, which is why work through on the board notes are not that helpful. Apparently, I agree to teach classes every 2 years. But the way that this is intuitive for me is that you can do the same sort of, you can combine the kind of 2 different tools that we already worked out. So we know that if we had a 1D model, we could sweep through upstream to downstream and define it as a series of conditional probability statements. And we also derived what's the joint probability of that row. And we already saw that that's this. And so then you can think about it as you do the joint probability of this, and then you sweep upstream to downstream through the columns. So you're kind of combining 2 of the tricks that we derived earlier. Where you say, this one has a certain joint probability, and this one has a certain joint probability given this, the prediction given this. This one has a certain joint probability given this. And that's sort of, I think, when I was doing that, that was the sort of most intuitive way for me to start thinking about this. So, basically I replace the scalars that I had before with the vectors of this epsilon matrix. And just say, the epsilon for row I plus 1 is equal to, it follows a conditional probability with an expectation equal to the row before it times this row parameter, and you just sweep through the factors of that matrix the same way. And instead of this scalar sigma squared, we're swapping in the epsilon that we already derived for each as the covariance of each row. So the key here, I guess, the reason I think that this is a useful way to think about it is you can start seeing that you can build up multi-dimensional smoothers just from, the same tricks we've been doing for unidimensional smoothers. And, the big spoiler for next week is that when we do spatial temporal models, we're going to do it the same way again. So, you can define sort of the joint probability in two-dimensional space using a bunch of tricks, and then just sweep across time the same way. If you wanted to have, space and time, and, I do multi-species stuff, I do the same thing in a fourth dimension, and you can do this kind of ad infinitum or until you run out of computational power. So, the spoiler, I guess, is that we already have all the kind of computational conceptual tricks we need to do spatial temporal models. The code for this week and next week is essentially showing ways of coding it in fewer and fewer lines, so that once you start doing spatial temporal models with 4 dimensions or 5 or 6, you don't have to do a bazillion loops. You can kind of do a smaller, code base, so it's easier to read. But, otherwise it's conceptually pretty similar to what we've been doing.
Why is, I think I missed the punchline, why is the matrix still 3 by 3? I was expecting, it's just for one row, is that what?
Well, yeah, sorry, yeah, so epsilon I is just one row of the matrix, right. Capital E.
Right.
So if we've got this matrix with all 9 points being a 3 by 3 E, capital E matrix, the way we're doing it is we're just sweeping upstream to downstream through the rows. So we pull out the vector in this one row and say it's conditional probability given the row above it.
So when you show that 3 by 3 matrix of ones on the diagonals and then row squared, is there like a row for every other column or yeah?
Yeah, and so so we'll get to that in a second.
Okay, that's
yeah. So and I apologize. I remember having this problem before where row, the Greek letter, of course, is the common term for correlation. And of course, when we talk about matrices, we're going to talk about rows and columns. So people have to guess by context which row I'm saying. And just ask me, I guess, if you get confused.
Isn't the way that it's kind of written, it ignores the conditional probability between different rows? Or between different columns.
Yeah, so you mean like what's the correlation? Are you saying what's the correlation between epsilon I minus 1 and epsilon I plus 1 or something? Like if for non-adjacent rows, how it's dealing with the correlation?
No, or wait. Oh, okay, never mind.
We could get through a few more slides of kind of the joint specification of this whole process and then come back to some of these questions. How do we do that? And then, so without really trying to derive it, the claim that I'll make is that so epsilon is a matrix, and so if you want to take that matrix and compress it to a vector, the math notation for that is this operator vec that just takes something and vectorizes it. So the left-hand side, vec E, is a matrix, a vector of length 9, and you might wonder what's the joint probability of this whole vector of length 9, given the way we defined it before, and that joint probability looks like this. So if you've got so this block is the first row of that matrix E, and this is just the joint covariance that we kind of worked out by hand before. And if you go one row down, it has, the vector epsilon I plus 1 is equal to epsilon I times rho. So it makes sense that the next row down is going to be rho times that correlation. And if you go 2 rows down, it's rho squared, kind of using the same logic we had before. And so that, that's sort of how you can see that it would look something like this. I'm not going to talk too much about this, except that it's the same conversion power series that it converts from the conditional variance if you go 1 step to the marginal variance if you go kind of an infinite number of steps. And so anyway, so hopefully this sort of has some, or do people want to talk about this notation a little bit more? I mean, the point is that this generates a covariance matrix for this vector of length 9, because it's a 9-length vector. The joint covariance has to be a 9×9 matrix. And this is just sort of a block way of describing a 9×9 covariance matrix. So people, these sort of block matrices come up often enough that people, want to come up with sort of algebraic notation for it. And the algebraic notation for that is the Chronicle product. So sort of the second big picture point conceptually of this that's probably new to people is the Chronicle product. I mean, if people got into within linear models, you can start getting Chronicle products running around, but it's not usually something that people feel like they need to teach unless you get into spatial temporal models, I think. So maybe raise your hand if you've seen if you've sort of used a Chronicle product or
only for the spatial temporal models.
Yeah. Okay. Yeah. But the point of a Chronicle product is it's doing a block. It's a matrix exponential, or it's called an outer product, where if you have a matrix, a 3×3 matrix, and the Chronicle product of that and another 3×3 matrix is a 9×9 matrix. And so it kind of multiplies the dimensionality of those matrices. And specifically, it assembles a matrix where you've got the second term of the Chronicle product in these blocks multiplied by just each element of the first matrix. And so again, if you've got a 2×2 matrix times the Chronicle product with a 4×4 matrix, you're going to end up with an 8×8 matrix, where you have the second matrix repeated, but multiplied by the elements of the first matrix. So one way to think about this joint matrix, kind of given that definition of a Chronicle product, you can see that this matrix that we're assembling here is just the Chronicle product of the correlation matrix and itself. So this, this top right row element is rho squared, and that's the same as the rho squared in each block. The diagonals are all 1, and that's the same as the outer part in this form. And so you can see that this covariance, sorry, that's actually not, is technically it's the correlation, it's the correlations times the marginal variance. So sorry about. Well, I can finish fixing that notation later. Any questions while I try to do this? Fix this typo. So can you read the question to us?
So this is just a matrix for 2D space? Which matrix?
Let's see. So, yeah, Kristen, you mean that this sigma total is the matrix for 2D space? Is that your question? Yeah. So correct. So the way I'm defining it here, sigma total is the Chronicle product of of the correlation for one dimension and the correlation for the other, and then times their marginal variance. If you had 3 dimensions, you could also do a Chronicle product of matrix A and matrix B and matrix C. Or if you had 4 dimensions, we'd eventually get to the point where we could have a Chronicle product of A, B, C, and D, or whatever. So this sort of Chronicle product definition of the joint covariance matrix is the helpful way. It's one way of kind of compactly writing out math notation for high dimensional smoothers. And that's the reason that people want an operator that represents that.
So I think this is theoretical, but if you had, if A, so you're saying A and B are identical in our case, but if A and B weren't identical, would that be saying that you think that the correlation between like latitude is different than the correlation in longitude or something like that? Does that make sense?
Yeah, totally. So you could have, I'm sorry I didn't catch that typo before, and of course it'll like propagate through a little bit, but yeah, so you could have one R matrix look like this correlation, and you have a row, a parameter row that represents the correlations across columns. And then you could also have a separate correlation matrix with a separate row that represents the correlation across rows. And then you could take the Chronicle product of those 2 different correlations, and you'd end up with the covariance of that joint process. Where you're estimating a separate correlation across one axis than the other axis.
Cool. Yeah, that's like useful when for when you mention like if you had like 2D space and then a 3rd dimension being like age or something.
Totally.
Like that would be kind of necessary. You wouldn't want to impose the same correlation. You would
totally
something that wasn't true.
Yeah. Yeah, and so next class we'll talk the notation is written out like to involve a little bit about geometric anisotropy. So we haven't really used the term isotropy or anisotropy yet in the class. But isotropy is the idea that you go any direction and you have the same decorrelation. So like, in small distance, in space like here, like at small scales, that's kind of a reasonable assumption oftentimes that, locally, if I move a certain distance, the temperature of this room will change kind of the same amount or whatever. But at large scales, like in my fish example, if you're going off the shelf, you decorrelate much faster than if you go along like common depth. The same thing I'm sure occurs with the public health stuff. And so if you have the decorrelation rate depends on the direction you're facing, and that's sort of a special case. Assuming that those rates are constant across your whole domain, so anytime you walk this direction, no matter which way where you are, it's the same decorrelation rate. And assuming the same, that is the same across space, that's defined as sort of geometric anisotropy. We'll maybe get into that more on Thursday.
Does it matter which dimension you write first? That's the question.
Yeah, so if you write it out this way, it's just bookkeeping, really. So let's pretend you've got, instead of a 3x3 matrix, we've got a 3x4 matrix. And let's just talk about the case where the decorrelation rate is different for rows and columns. So we've got an A matrix that's a 3x3 matrix and a B matrix that's a 4x4 matrix. If you switch the order of the A and the B matrix in the Chronicle product, it would just, the only thing you'd have to change is you'd have to make the transpose of the E matrix to make sure that the elements of E correspond to the right components of that Chronicle product. And I guess that's as far as I want to get into that. It's, again, the point, I label that as bookkeeping because it's sort of an issue of making sure that the way you're using your vec operator plays well with the order that the Chronicle product is doing things. Yeah, so the other benefit of the Chronicle product, and I wish I'd caught this notation problem before, is that the matrix inverse cuts through it. So if you can take the Chronicle product, if you want to take the inverse of a matrix, you can define it as the inverse where you do the Chronicle, you replace this matrix with the Chronicle product, and that's equal to doing the Chronicle product, the inverse of each component of the Chronicle product first. I'm not going to mess with the notation on the fly here. I'm sorry. Because that would just be too confusing. So in this case, this is a 9 by 9 matrix, and so inverting it requires a bunch of extra computations, whereas it's much more convenient to invert each of the 3 by 3 matrices individually and then take the Chronicle product. And furthermore, we know what the inverse covariance matrix or the precision matrix of each of these AR1 processes is already. We wrote it out. We derived kind of tried to have kind of an intuitive reason for why it works last week, and so we can write out that individual covariance matrix by hand, and then we can just take the Chronicle product of those inverses and never have to invert the joint matrix. So writing this out, that each individual component, each row or each column has a precision matrix that looks like this, that's sparse because it's this, the partial correlation is 0 for locations that are not adjacent to one another. And so if we do the Chronicle product, we get that the inverse looks like this. And, where again, it's just, it's zeros on the off diagonal, or that, anything that's not adjacent in the block. So this whole block is 0. This whole block is 0. And then within each block, there's a bunch of zeros as well. So you can start seeing that if you take a Chronicle product of 2 sparse matrices, you end up with something that's kind of even sparser. So specifically, sparseness is multiplicative in this Chronicle product. So if you have an A matrix that's NA by NA, so a square A matrix, but it has NA non-zero elements, so defining sparseness is NA divided by the total number of elements, and you've got B doing the same thing, then the Chronicle product has sparseness equal to the product of those two. So if you've got one matrix that has only 10% of cells that are non-zero, you've got another matrix that only has 10% of cells that are non-zero, then their Chronicle product will only have, 1% of cells that are non-zero. So it scales attractively where, the problem kind of keeps getting easier the more, relatively, the benefit of using sparse matrices gets greater the more dimensions you're working in. So, INLA uses sparseness, STAN does not use sparseness, and TMB uses sparseness. And so you can start kind of seeing intuitively that, STAN scales relatively badly for doing, multidimensional spatial temporal models because the benefits of doing sparseness get greater the more dimensions you put in. So I think that's all the content I wanted to cover today. And unfortunately, we don't have a whole ton of time to look at the code. So can people see this, or maybe I turn off? Can people see that okay? So once again, what I want to show is that there's several different ways that you can code this in TMB, and all of them end up being about giving almost the same answer, except for kind of boundary conditions that I want to put in because I want to keep the example simple. And they all work at about the same speed, so TMB, because it's a compiled language, is able to end up with about the same speed in a number of different ways that you code it on the C++ side. And so the goal is not to show sort of the benefit of having a separable model versus a non-separable model. It's just kind of to show that there's a bunch of different ways you kind of arrive at thinking about the same spatial process. So first I'm going to simulate data where I generated a 10 by 10 grid, I think. So there's this loc xy, that's a data frame, 100 linked data frame, where the first column is the x coordinate and the second column is the y coordinate, hence the term loc xy. And I'm going to define, I'm going to use this Rmx function from the package random fields, where, let's see, I forget what I think it's a random model, that's probably what that stands for or something. And exp means that it's an exponential function. And if we had time, I'd love to switch this code over to use, concepts we can do this ourselves without using the random fields package. But for now, we'll just use the random field package to simulate this random effect epsilon xy. That's the 10 by 10 matrix of the random effect. We then simulate counts from this log-linked Poisson generalized linear mixed model, where the sum across all 100 cells is 1,300, and we end up with this matrix CXY that's 10 by 10, representing the counts at each location in this 2 by 2 grid, two-dimensional grid. I'll also use these M matrices, and if anybody's interested, this is how you can generate sparse matrices in R. So in this case, M0 is this diagonal matrix. M1 is a first-order autoregressive. Well, it's one for any cell that is adjacent to another cell, and then this M2 is one if they're separated by diagonal cells, and 0 otherwise. So, and we'll use those later. Well, M2 is. So, just to sweep through the R code, because we don't have a total ton of time, we have a single CPP file that we'll look at again. We run it, and all of them take less than a second to fit this 100 length random effect that's correlated across 2 dimensions. The second way takes a little bit longer than the third and the fourth, but they all take within 1 to 2 times as long as each other. So there's not a huge speed difference. And we can confirm that all of them correctly detect sparseness. So the version 4 has a sparse matrix Hessian, version 3 does, version 1, I'm calling it. I'm not sure where the other one went.
I think you need to do dev new.
Oh, I need to do dev new before it or something?
I think right now.
Well, no. It doesn't, yeah, it doesn't matter. Version 0, which was missing, also looks exactly the same in terms of sparseness. So this this banded Hessian, this internal one is well, you can actually see that this follows that block structure that you expect with a Chronicle product. So this block is one row, and this block is the next row, and this block is the next row, and then this one is a row with the adjacent row. And so, for instance, if you count, this cell has a non-zero Hessian for adjacent, well, let's take this second element here from the bottom. It has a non-zero partial correlation with two cells that are adjacent to it within the same row. And it has a non-zero correlation, partial correlation with the three cells that are one row down from it. And similarly, if you go up to this one, it has, you know, the adjacent ones in the same row, and then a partial correlation, non-zero partial correlation with the three cells, one row up, and a partial correlation with the three cells, one row down. So, you know, knowing something about Chronicle products should allow you to start interpreting sort of the structure of some of these Hessian matrices that you can get out of T of E to, you know, actually infer something useful about the structure of the model. And then looking at the C++ code, it has very similar structure to the lab code that we did last week, where we've got these sort of helper functions put in here, so that isNA is sort of a hint that I thought maybe we would do an exercise where there's some cells with no data, and so we could put in that data matrix that's like Cij and put in some NA values, and that would mean that when doing the likelihood calculation for the data, if it found an NA, it would just skip that, but it would still predict the random effect of that location. We could get predictions at unsampled locations that way. Kind of similar to how we did the homework 3 or whatever it was. And then we've got the same functions that I built for the log determinant and the DMV norm that we saw before. And then in the main block of code, we've got the sort of options. You know, we've got just the read in the data. We can talk, let's come back to the M0, M1, M2 in a second. But we read in the data, we define the parameters. Then we've got a block of bookkeeping that transforms parameters and calculates indices and sets up the joint negative log likelihood. As always, and then we have this block of code that defines the probability of the random effects, and that's the biggest block of code here. And then we've got a really short block of code that defines the probability of the data given the random effects and fixed effects. And we've got the outputting, so kind of bookkeeping again at the bottom. And so what's, you know, similar to the structure of the code before, we've got one block of code where we do this, we do it one way, a second way, a third way, and a fourth way for doing the probability of the random effects. So walking through those, let's see, the first one, the first one just generates the precision matrix by hand. So I define a precision matrix. QIY is the precision matrix for a single row. So in the 3x3 case, it'd just be a 3x3 matrix. In this case, we're going to be fitting a 10x10 data set. And so QIY is just a 10x10 matrix for the joint probability for a single row. The joint precision matrix for a single row. And then down here, we are generating 2 of those precision matrices. Let's see. Yeah, we're generating 2 of those precision matrices. We're vectorizing. How does this work? so temp YY, it ends up being kind of complicated bookkeeping, but temp YY is just the predictions given the row before it. So it's just taking this and making a vector of predicted values. And then defining the yeah, I'm sorry. How are we doing this? I remember this. Okay, so this one's the simplest way. So okay. So here we define the precision matrix. Q0YY is the precision matrix for the first row, given the marginal distribution, kind of like the starting condition for last week, where we had to define a probability for the first location, the most upstream point, and then we define all the downstream points given the one that's one upstream from it. So Q0YY is the matrix now for that first row, and Q1YY is the precision matrix for a row, given that you have a row upstream from it. Similarly, temp Y, if you're doing the first row, temp is just the prediction where the first row is epsilon is that vector, and if you're sweeping downstream, the mean is just equal to your random effect minus the prediction from the last one.
So this is where if you had more dimensions, it would be like really ugly. You'd have to like
it would start getting really ugly.
Initialize another dimension, basically, right? Because you have to initialize twice because you have two dimensions.
Totally. Yeah, exactly. So this way only really works for this two-dimensional case. And I'm sorry that I'm traveling right after class, so I didn't look at the CPP code quite carefully enough. But this is the way that we're kind of defining it here, where we do the probability of this first row and then the probability of the second row given the first. If we had a third dimension, I have no idea how we would do that. So next, we can take the precision matrix for a single row, define the Chronicle product of it, and then there's this extra sigma squared because of the mess up in notation I had, where you really want to take the Chronicle product of the correlations and then just put in the marginal variance once. In this case, I've got the variance in each of these components, and so I've got it put in twice here. So it would work either way. It would just define the definition of sigma squared would hold across models without correcting for that. But anyway, so QZZ is the Chronicle product of two of these matrices. So QZZ is a 100 by 100 matrix, and it's dense because I didn't define QYY to be a sparse matrix, so it doesn't know that it could be a sparse matrix itself. So if you remember, this second option, which is called option 1, is the one that takes the longest of these options. It still detected sparseness, so it's still pretty quick, but assembling this dense precision matrix is a time suck, and so it ends up taking about 3 times longer than the most efficient way we could have done it. But it's still more compact code than the first way, and you can also kind of start seeing how this one could scale in more dimensions, because if you've got a 3-dimensional model, you could just take the Chronicle product of one matrix and another, save that, and then do the Chronicle product of that same matrix in the 3rd dimension. And so this one would extend to higher dimensions, whereas the first one wouldn't. So the joint precision matrix is assembled from this Chronicle product, and then this loop is just me doing the vec operator. So I'm taking this matrix epsilon and just reducing it down to a single vector, and I'm calling epsilon z. And then I do this big joint multivariate normal with this vector and its precision matrix. The third way to do it, which is by far the most, it's simultaneously almost as fast as the fastest method, and it's only one line of code, but it's sort of difficult to read code if you're not used to it. So, and it's using a bunch of these caps locked functions that are in package namespace density, or yeah, namespace density. So I can just put that down here, actually. So we started seeing namespace density in the code last week. Do I remember right that I didn't have that much time to talk about it?
We talked about scale in AR1.
Yeah. So hopefully we did, because here we're kind of taking the same thing and just adding a new, the third, I think these are well, there's only 4 functions we'll ever actually use out of namespace density. So one of them is this GMRF that I'm kind of commented out, and you could look at how the GMRF does the same thing if you wanted later. Well, no, we'll use GMRF in a second. So GMRF is 1, AR1 is the second, Separable is the third, and Scale is the fourth. And I promise those are the only 4 kind of pre-built functions that I think we'll need to use. So AR1 takes as input a single parameter row, and it returns this sort of unit normal, or kind of variance of 1 AR1 process with the input row. And then Separable takes 2 of these different processes and basically takes the Chronicle product of them and just makes a separable correlation function that includes both dimensions. And that term separable, there's sort of this tradition in space-time models of saying that any model is separable if you can just calculate the correlation as the product of the correlation of the 2 dimensions individually, which is sort of what the Chronicle product is doing. So that's sort of the reason that this function is called separable. So now we've done this separable combination of 2 different AR1 processes, and then it only has had row input into it. So we have this int that is missing the kind of variance parameter of the process. And so then we use the scale function the same way we did the other class, where we have to calculate what's the marginal variance of this whole process, and that's this thing. So this is the rescaling of the variance of the process. Yeah.
So if you were going to have, never mind. Yeah, sorry.
You sure you don't want to? I mean, I don't know.
No, you said you'd go into this next time, so I'm just.
Yeah. So next time we'll get into kind of unequally spaced locations. Okay. So anyway, so the net effect of this is that this returns a function. Well, it returns an object that computes a function. This separable combines these to return a function that would generate, calculate the negative log likelihood for this separable process. Scale returns a function that does the same thing but has been now rescaled. And so at the end of this, we just have this input epsilon xy, which is technically a matrix, but the function is smart enough to just coerce that to a vector internally. So this line is returning the joint negative log likelihood, and so you do the plus equals instead of minus equals like the rest of our, most of our functions. And then finally, the last way we can do this is using sparse matrix computational methods that we can access from the package eigen. And it uses these inputs M0, M1, and M2. So M0, M1, and M2 are me, having seen that this Chronicle product has this particular structure where part of it's this diagonal across the whole matrix, part of it is the off diagonal, and then part of it is the cells that are they're diagonal from one another. And so we we saw that structure in the Hessian when we plotted it. So this Hessian structure has has this particular structure. And we can hardwire that calculation by using 3 matrices. So if we plot, sorry about this, we're out of time, but I kind of want, I'd love it. Can people stay another 5 minutes or? So in this final case, I've defined a sparse matrix that's just the diagonal part of that Hessian, and then another one that's the one cell away, and another matrix that's diagonals. And so if you do M0 plus M1 plus M2, you get the Hessian structure. You get the banded structure of our original Hessian matrix. So kind of, you know, algebraically, I kind of know the structure of this model. I can put in a sparse representation of it that I calculate, I pre-process in R, and define these sparse matrices M0, M1, and M2, and then I can pass them to the C++ file using this input data sparse matrix. So data sparse matrix is this, you know, is the term telling C++ that on the R side, it's going to hand it a sparse matrix. And then I assemble the precision matrix, where I define QZZ as a sparse matrix using package eigen. I assemble it from those 3 different pieces and the parameters, and then I can input it using this function GMRF that's built into T and B, where it just expects the precision matrix of some big process, and then you give it the vector of your random effect. And so ultimately, this one is sort of, you know, probably you know, if you can get a model to the point where you can actually work out the sparse matrix structure of it and just pass it directly, this is probably the best way to do it. I kind of think this is the next best. And I don't like it quite as much because it does all this like computation on the C++ side where it's internally generating those matrices, whereas this one I can do it on the R side once and never have C++ do it again. But in practice, for this size of model, those two last methods both took, well, one took 0.212 seconds and one took 0.11 seconds. So they're about the same speed.
Is there a way, it seems like for like a 2-dimensional case that the code you use would always work for, right? Like for generating the 3 entities that you need.
Yeah, if they're equally spaced. Yeah,
Okay.
if it's equally spaced locations.
And I guess it would be more complicated if it wasn't symmetrical like it is.
Yeah, exactly.
I was just wondering if there's like a way to like programmatically come up with a function that could do that for however many dimensions.
Totally.
I'm sure it's doable.
Yeah, for sure it is. And we'll see on Thursday that INLA, the punchline for Thursday is that you can define this, the precision matrix for unequally spaced locations in 2 dimensions as the sum of 3 different sparse matrices the exact same way. So INLA is essentially using that 4th approach, that M0, defining the precision matrix as like M0 plus M1 plus M2 and some parameters. INLA is doing the exact same thing, except it just can define the M0, M1, and M2 matrix for any locations in 2D space. And you know, there's a really complicated paper about finite element analysis about why that works that you don't really need to know because the package INLA does that automatically for you. So, okay, so we're out of time. Jay will be teaching on Thursday. Yeah, and I'll see everybody again next Tuesday. The final lab exercise, well, the final homework that's not related to the project is due the coming Tuesday. And it's a 1D spatial nonlinear model. So feel free to email me. Thanks, everyone.